Hi, in this lecture, let's look at the reabsorption that occurs in the proximal convoluted tubule. We've done filtration, now let's talk about reabsorption. So this is, should look pretty familiar, and this is sodium reabsorption in the proximal convoluted tubule. So again, I'm just calling that the PCT for proximal convoluted tubule. And this is the sodium reabsorption is done as a primary active transport. On the basolateral membrane. Primary active transport. Remember that means if it's primary that ATP is required. ATP hydrolysis is required for primary active transport. And again, this is going to occur on the basolateral membrane. Which will identify abbreviate membrane. So this membrane is on the basal surface. Lumen is to the left. And this would be the interstitial fluid, the ISF, or the extracellular fluid, let's call it the ISF. So this is on the basal surface and the lateral surface. These are the cells that are sitting on the basement membrane of the epithelium that's lining the tubule lumen. This membrane is on the luminal side, or the apex of these epithelial cells. So this is the apical membrane, apical for apex. Okay, apical membrane. So it's good to note where these transport processes are occurring on the apical side, on the luminal side, or on the basolateral side to the interstitial fluid. And that's about it for this figure. There is the sodium that's in the filtrate, and one of the first things we need to do is reabsorb the sodium so it enters the cell by facilitated diffusion. Just straight down a gradient. Should look familiar to you. And then it gets pumped out by the sodium-potassium pump. And this text, along with others, refers to the sodium-potassium pump as a sodium-potassium ATPase, because it pumps sodium and potassium while hydrolyzing ATP. So the sodium-potassium ATPase, again, is also known as the sodium-potassium pump. Okay, so that's the first step, is to reabsorb the sodium. And that creates a sodium gradient that we can then use for secondary active transport. So this slide is showing sodium-linked glucose reabsorption in the proximal convoluted tubule. Again, this story is all in the PCT. Remember, that's where about 65 to 70% of all reabsorption occurs, is in the proximal convoluted tubule. So here we're reabsorbing sodium and glucose. So this process of sodium-linked glucose reabsorption is secondary active transport. Again, we're staying in the proximal convoluted tubule for this entire lecture on reabsorption in the PCT. So what this shows, and should look familiar, is, remember, we just created the sodium gradient previously, so we're still out here in the lumen, and now we are trying to reabsorb the glucose. And the sodium diffuses in, and at the same time through this channel in comes the glucose. And this transporter is often referred to, or it's abbreviated as an SGLT, which stands for sodium glucose linked transporter. Oops. Sodium, glucose, they're linked together. This is secondary active transporter. So the glucose reabsorption is linked to the sodium. And it brings glucose in against its concentration gradient, where glucose is low in the lumen and high in the cytosol. So to bring it in against the gradient is going to require energy, and the energy comes from the sodium moving down its concentration gradient. We spent some time on these transport processes in biology, or in A&P1, and in Biology 160. So to get rid of the sodium, this looks familiar. The sodium is just removed by the sodium-potassium pump. Burning ATP, potassium in, sodium out. We've gone through that 100 times. The glucose then goes out by facilitated diffusion. It's going down its concentration gradient from where it's high inside the cell to where it's now low on the outside. And that goes out through a GLUT transporter, or a glucose transporter, which is just facilitated diffusion. So I'll just connect these two and you can just read the key in the figure. Okay, so we've gone through that before. This should be review, reading the numbers, sodium moving down the gradient as we looked at through the SGLT, which just stands for sodium glucose linked transporter. Protein pulls glucose into the cell against the gradient, and then glucose diffuses out through the GLUT, which is just a glucose transporter, and then the sodium potassium pump is continuing to pump sodium out and potassium in, creating that gradient. So let's move on and take a look at other transport using secondary active processes. So this we could also use as a practice to show the reabsorption of sodium, glucose, and amino acids. And this is all going to be done by, at least these two, are going to be done by secondary active. And sodium is going to be done by primary active. Hydrolyzing ATP. So let's get set up with a couple of cell membranes. Let's have some room to draw. So there's one cell in the middle of 3, and this will be our luminal side. And this will be our extracellular fluid, or interstitial fluid. And we can put in a blood capillary if we like. And this would be a paratubular capillary. Remember, we're in the kidneys. Have to get it back into the blood. So let's start with sodium. First thing we have to do is reabsorb sodium. So some of that just leaks through little diffusion channels. And the sodium enters the cell down its concentration gradient. And then let's put in a sodium-potassium pump. And this is hydrolyzing ATP, so we could just write ATP here to remind us that ATP needs to be hydrolyzed. And then the sodium, let's just follow this out and get the sodium-potassium pump working. And then the sodium enters the capillary. And this is also moving potassium inside the cell. We've done a lot of that in A&P 1 and Biology 160. So that's primary active transport of the sodium. So there's the sodium, we're done with that. We've reabsorbed the sodium from the lumen on the left into the paratubular capillary on the right. And again, it entered this the cell by facilitated diffusion. I'll abbreviate. Facil-diff. Across the apical membrane. The sodium-potassium pump is on the basolateral membrane. And then let's reabsorb the glucose. So as we did before, let's put in a little transporter And I'll go ahead while I have the eraser out, I'll make another one for the amino acids. So there are multiple transporters on cell membranes, and they're generally quite specific for chemicals, molecules. So this is going to be secondary active process for these two. Secondary active. So you know that that means it's going to require ATP hydrolysis. So sodium enters again down the gradient. In comes the sodium. It is removed from the cell by the sodium-potassium pump that we already looked at. So all the sodiums kind of go together in this diagram and are eliminated by the pump. And we are also bringing in a glucose. So in comes the glucose. We just did this. So this was our SGLT again, if we wanted to abbreviate. And the SGLT again stood for what? S for sodium, G for glucose, linked transport. Okay. And then the glucose will leave through a channel on the basolateral membrane. So there's the glucose, and we've reabsorbed glucose back into the ECF, and that returns to the blood as well. And this process was facilitated diffusion, so I'll abbreviate that as FACDIF. And the amino acids are reabsorbed in a similar way, just by secondary active transport using a different transporter. So this basic story again is that it has to be linked to sodium. So sodium enters the cell. Get rid of it as before. And then we bring in amino acids. Which I often just abbreviate as AA. And then those amino acids leave through the same old pattern of facilitated diffusion. And they also enter the blood. So all this is occurring in these cells of the proximal convoluted tubule. So we have lots of carriers on the cells in this region of the kidney. And then what about water? We need to reclaim water. Remember that we are reclaiming the ions and the osmolarity stays the same. So if we're not going to change the osmolarity, we need to reabsorb water also. 65%, 70% of that volume is reabsorbed in the PCT as well. and we move water across the membrane efficiently through aquaporin channels. And in both cases, the water is just moving down its gradient by facilitated diffusion. It's basically following the sodium. So these are aquaporins. And the water just follows the sodium. And that gets picked up by the peritubular capillaries also. So, it's not bad. You should practice this until you really can show primary and secondary active transport without any trouble, and then the facilitated diffusion. So, secondary active transport is needed to move substances against the gradient, and facilitated diffusion just allows them to flow down their gradient. So that takes care of sodium, glucose, amino acids, and water. Let's look now at the reabsorption of bicarbonate. So I've already drawn a few of the transporters in this story. So again, this is occurring in the PCT, and we're reabsorbing the bicarbonate, one of our principal blood buffers. So let's start with sodium. Sodium is key to much of this. So again, here is the sodium. And this is using a different transporter, as you'll see in a moment. This one is antiporting hydrogen ions out of the cell into the lumen. So that's going to have an effect on pH anytime we're moving hydrogen ions around. And we'll leave the sodium-potassium pump alone for a moment. You know that it's going to be working on the right to remove the sodium, but let's get back to the filtrate. We had the sodium, and now let's put the bicarbonate in here. This is what we're after. This is what we're trying to reabsorb. And it would be so much easier if we just had a transporter to just reabsorb the soda or the bicarbonate as it is, but that's not the way it works. So the hydrogen ion combines with the bicarbonate, and you know what those two make. This is our same old reaction that we've done many times. That forms carbonic acid. And then there is an enzyme present on the apical surface of these epithelial cells that is going to break carbonic acid down into water and CO2. We've seen this many times. And that enzyme, of course, is carbonic anhydrase that I'll abbreviate. Okay, so we've taken the bicarbonate, broken it down into water and CO2, and the CO2 is then able to diffuse through the cell membrane by simple passive diffusion. And then we just do the reverse. Remember, we're trying to reabsorb bicarbonate. Well, there is no bicarbonate in the cell, but we have the CO2 in the cell. So again, it combines with water. And in the presence of carbonic anhydrase, which is also inside the cell, that produces the carbonic acid, which then splits. So I'm going to skip the carbonic acid piece for reasons of space and just go straight to the ionization of the acid. There's the hydrogen. And we have the bicarbonate. Makes sense? Brought the CO2 into the cell, combined with water. Carbonic acid was dissociated into the hydrogen and the bicarbonate ion. Now we have the bicarbonate ion inside the cell. Let's get rid of the hydrogen first, though. The hydrogen gets pumped out by this antiporter with the sodium. So the hydrogen just kind of goes around and around in a circle, being pumped out, coming back in, getting pumped back out. And the bicarbonate then exits the cell by facilitated diffusion. We have HCO3 with a minus on it. And that goes back into the cell. And then finally, we can deal with the sodium. And the sodium exits. And we are bringing in potassium again. So that's how we reabsorb bicarbonate ions from the lumen back into the blood. So start with the bicarbonate in the lumen and move that into the cell in the form of CO2. So you have to convert bicarbonate to carbonic acid to split to get the CO2. That enters the cell, you're back to bicarbonate, and then that leaves. And we've reabsorbed the bicarbonate in the cell. The other step to remember is what happens to the hydrogens from the carbonic acid when it ionizes. The hydrogens get pumped back out sort of at step 1 here with the sodium proton antiporter. So let's just put a note here that this is antiport. Secondary active antiport. It's secondary active because it's relying on the sodium again, diffusing down its gradient. Okay, well, what this should impress upon you is that none of this reabsorption can take place without a series of membrane transporters. So that takes us into the next couple of slides as we wrap up this lecture. And this picture, it's an interesting graph, it's useful. And what we're looking at here is the saturation or the transport maximum of a substance across a membrane. So if this is a cell again, here's the basic idea that we're looking at. If we're going to be moving any substance across the membrane, either one way or the other, it has to go through a series of some sort of transporter protein. On the apical surface, it has to go through, and then it has to go through on the other surface as well. So it's these transporters that we're looking at. And they have limits, is the idea. They can only transport so many molecules so fast. They don't operate at light speed. They have limits as to how many they can move per second or per minute or per hour. So here's a graph that sort of looks at that. So the two axes that are shown is the plasma substrate concentration. And you're probably already have figured this out from chemistry, but if you see anything in square brackets, it's one way of denoting concentration. So it's the concentration of some substrate in the plasma. And units appropriate, milligrams per milliliter. How many milligrams per milliliter? Maybe it's glucose. Maybe it's 200 milligrams per, be quite a bit. Maybe it's 20 milligrams per milliliter of glucose. But that's the plasma substrate concentration. And the substrate is just what's being passed across the membrane. So you're moving a substrate, let's say, from the lumen into the interstitial fluid. On the vertical axis is the transport rate. How many of those molecules can be moved per minute? So this is the transport rate, and rates usually have time in them. So, we can move maybe 20 milligrams of the substrate every minute across. So this number 2 would be 2 milligrams per minute, 4 milligrams per minute, etc. 6 and 8 milligrams per minute. So that's just the units, just explaining the axes. And again, across the bottom, this would be 2 milligrams per mil, 4 milligrams per mil, etc. 8 milligrams per mil and 10 milligrams per mil. Okay, so now what the graph shows is that reabsorption, so transport is reabsorption, because we're transporting it across the membrane. So this is reabsorption. So if we have, according to this graph, if there is 2 milligrams of substrate per mil here, how much of that gets transported? Well, it just so happens that it's 2 again. So we can, the membrane, we can move 2 milligrams per mil and reabsorb it. So we can reabsorb that, and the same with 3. So at 3 milligrams per mil, how much is being reabsorbed per minute? Well, it's about 3. Okay, let's go to 4. Same idea. You just go up, hit the line, and if we have 4 milligrams per mil in the plasma, we can still reabsorb 4 milligrams per mil, and we are transporting 4 milligrams per minute. What happens now if our substrate concentration, our plasma concentration, our plasma load gets too high? So let's go to 5. And if we have 5 milligrams per mil in the blood, in the plasma, the little transporters on the cell membrane are already maxed out. They can't absorb any more than 4 milligrams per minute. That's it. So that's the transport maximum of this system. So transport maximum, TM, is the transport rate at saturation. So those little carriers, these little protein carriers on the cell membrane, these are maxed out. They can't work any faster. So in this case, the transport maximum equals what? 4, and then what are the units? We're talking about transport. So it'd be 4 milligrams per minute. And that's the transport maximum, the TM, for this example. It doesn't matter how much is in the plasma. We can go all the way up to 10 milligrams per mil. Way out here. And if this is glucose, for example, we can still only absorb 4 milligrams of that substance per minute. So what happens to the rest of it? And that takes us to the next figure. So this is a set of 4 figures. It's basically breaking down the idea that we had on the other side. So let's just go through them in order. A, B, C, and D. And look at the diagram, look at the axes and see what each graph is showing individually before you sort of get too excited about the whole picture. So starting with A, the whole story here is glucose. How does the glucose, or how does the nephron handle glucose? And you know that it needs to reabsorb the glucose. Because normally we reabsorb 100% of the glucose if our blood sugar is in normal limits. So let's take a look at this. What this shows is glucose concentration, okay, milligrams per 100 mils. So you should probably be familiar with a deciliter. So 100 milliliters equals 1 deciliter. Okay, this graph, or series of graphs rather, shows they're using the example of glucose. Glucose in the nephron. And you know that we have to reabsorb 100% of the glucose as long as it's in with normal limits. So it's just a series of 4 panels that breaks down the idea that we looked at on the other previous slide. So take it one slide at a time, or one image at a time. There are 4 of them here, and let's just work through it piece by piece, starting with the first figure, top left, panel A. It's showing the plasma glucose concentration in 100 milligrams per 100 milliliters. So you should probably be familiar with the term of a deciliter. So 1 deciliter equals 1/10 of a liter. Decimal for tenths. 1/10 of a liter, which is 100 mils. So in this figure, it's milligrams per deciliter. Now you can just say 100 mils if you like, but you should know that a deciliter is 100 mils. It's a common unit. Okay, so, and then this is just the filtration rate of glucose. So this is back at the glomerulus. Well, there's no limit on how much can be filtered at the glomerulus. And however much you have in the blood, it just gets pushed out. So if we have 100, 300, 500 milligrams per deciliter, we're just going to push it out into the filtrate. So that will be a lot of glucose in the filtrate. Remember, we have to reabsorb that. So then we go over to the second panel, and this is what we were looking at before. So this is the story with glucose. Again, units are milligrams per deciliter in the plasma. And how much of that gets reabsorbed. This is what we were just looking at. There's our TM. So this is the reabsorption for glucose. And it's approximately, what? What's the transport maximum for glucose, according to this figure? Well, it's there's the magic spot. At 300 milligrams per deciliter, we can reabsorb 375 milligrams of that per minute. So the TM equals 375, get the units correct, milligrams per minute. Okay, we did that in the previous slide. And it also marks out the normal range for glucose. So this is normal plasma glucose concentration between 100 and 200 milligrams per deciliter. So this is the normal range. And as long as our blood plasma level stays within that range, we should be able to reabsorb 100% of it, because the little transporters in the PCT have not hit their maximum. They can handle 200 milligrams per deciliter, which is here. So we reabsorb it all. Okay, let's go to C. Well, again, it's the horizontal axis is the same for all of these figures. It's just plasma level for glucose. But this figure is showing the excretion rate. And excretion is how much is excreted, removed from the body. And this, it starts at 300. So once we exceed the renal threshold, which was 300 milligrams per deciliter. So there's our renal threshold. We can't absorb any more. The kidneys can't. At that point, the excess starts to be excreted. So at 400, we have high blood glucose. We're at 400 milligrams per deciliter. How much gets excreted? Well, we don't really have numbers over here, but I figure out that it's going to be 100, and that's how much gets excreted. That's what's going to show up in the urine. And the fourth panel just puts all three of those together into one. So the red dots, again, are showing filtration. How much is being filtered out. And the green is showing reabsorption. And there's a maximum to reabsorption. That's the transport maximum. And Tm. And then the blue line is the excretion. So just a way of kind of showing kidney function graphically. So I'm going to stop here, and the next lecture we'll talk about the loop of Henle.